Hello everyone, this is Rohan Mishram, and in our previous lecture we have tried to shed light on topic like multiple sequence alignment, and we have tried to understand what are progressive strategies, and we have taken a typical example of cluster as a software that performs such multiple sequence alignment. Okay, so now you people are aware of what is multiple sequence alignment. Well, now let us move our discussion forward and try discussing what are the application of such multiple sequence alignment. So one of the most important application of multiple sequence alignment is to perform phylogenetic analysis for a given set of protein or nucleotide sequences. Okay, so that is one of the most important and popular application of multiple sequence alignment. And in this today's lecture or this today's video, our main focus of discussion would be to understand initially what are the basic concepts of these phylogenetics or molecular phylogenetics. To be specific, we would try and understand some basic terms and terminologies that are normally used in discussions wherever your evolutionary relationship among genes and species are concerned. So this is what we are going to discuss today in our bioinformatics class. Okay, so well, first let us start out with just defining what molecular phylogenetics is. So in broader terms, phylogenetics, you can define it as simply the study of evolutionary relationship. Well, say for example, if you have been provided with some multiple species, which includes rodents, primates, birds, crocodiles, marsupials, snakes, and lizards, your molecular phylogenetics or evolutionary relationship studies would give you an output like how all these species are related to each other. So that is the ultimate objective of molecular phylogenetics, as to how the species are related to each other, right? So just to begin our discussion, let us first discuss a few examples of what we have learned so far using the modern phylogenetic techniques. Okay, so we are now able to answer questions like which species are the closest living relative to modern human. With help of modern phylogenetics, now we know that which genetic changes are unique to human lineages and that are not present in its apes counterparts, right? Now we know using these molecular phylogenetics answers to questions like what was the most likely geographical location of the common ancestor of African apes and humans. And there are many and some countless other examples which are now answerable due to modern techniques like molecular phylogenies. Okay, so well, now we know that which species are the closest living relatives of modern humans owing to this molecular phylogenetic data. Okay, so well, in modern view of molecular phylogenetics, okay, on the basis of whatever sequence analysis we have done from mitochondrial DNA, most of the nuclear DNA encoded genes and some DNA hybridization experiment, all of these modern phylogenetic analysis indicate that the bonobos and the chimpanzees are more closely related to humans as than either of the gorillas or orangutans. Okay, while on the other hand, on the contrary, The pre-molecular view of evolution was it was like the great apes, which includes your chimpanzees, gorillas, and orangutans, like chimpanzees, gorillas, and orangutan. These form a separate clade from humans. Okay, and the common ancestor of humans was believed to diverge from the related apes some 15 to 30 million years ago. But now, in modern phylogenetic view, we know that the common ancestor of all the apes that we, or all the primates that we know, that is that they have diverged, say, some 14 million years ago, and not 15 to 30 million years ago. So with help of such mitochondrial DNA, most of the proteins and DNA sequence analysis, we are now in the position to answer questions as to how humans might have evolved. Okay, so these are, one of the most prominent application of such molecular phylogenetic analysis. Okay, so the objective of this lecture is to first let you know what are important terms and terminologies, what do they mean. Okay, and then we would try to learn how do we build up such phylogenetic tree. Okay, so first we would spend some time discussing as to what are important terms and what are important terminologies and how do we use them, right? And the subsequent next lecture we would try to have theoretical background behind how those phylogenetic trees are developed. Okay, so the first thing first that you should know is that the physical position of the different species in the tree is not very meaningful. It doesn't really matter where the uh, where the uh, where the sequences or where the species are located in tree. Okay, it is only the structure of the tree that would matter. Why? Because it is possible that one can swivel these trees wherever there are nodes available. Okay, these trees somehow can be swiveled at the internal nodes. Okay, so well, say for example, if you swivel your tree at this particular node, the topology of tree might appear to be different. Well, say for example, if you have got sequence A and sequence B, and if you try to swivel over here, what you would get is that this C would come down. Okay, and again you would have sequence D and E. Okay, so this tree is perfectly analogous to this tree. So that is what I want to demonstrate over here, is that the physical position of the sequences or the species will not really matter. It is only the structure of the tree that would matter. How these sequences are related to each other, that is what is needed to be properly understood from such tree. Okay, so let us try and understand some common phylogenetic tree terminologies before we proceed for further discussion. If you have been provided with any tree, the nodes that are present on the terminus of your tree are usually referred to as terminal nodes. So over here, the nodes that are present at the terminus or at the end of the tree, well, say for example, nodes that we have designated as A, B, C, D, and E, these are called as terminal nodes. And these terminal nodes usually represent different taxa. We call the terminal nodes as taxa. And this taxa can represent genes, this taxa can represent population, this can represent species, or say some protein sequences, it can be anything. Okay, so these taxa are used to infer phylogeny. The objective is to find out how these taxa are related to each other. So besides the presence of terminal nodes, your phylogenetic tree can also have internal nodes, or these internal nodes are also sometimes referred to as divergence points. So these divergence points represent the common ancestor of the terminal nodes. Okay, or you can say they represent the hypothetical ancestors of your terminal nodes. So over here, the internal node are represented by these white circles or white spheres. So this particular white sphere or this particular white internal node represent the divergence point between the terminal node D and E, and hence it can be called as the common ancestor of D and E. So besides these terminal nodes and the internal nodes, if the tree is rooted, we have something called as the ancestral node or something called as the root of the tree. Okay, so these are different nodes that are present in your phylogenetic tree. Your phylogenetic tree can contain root or ancestral node that represent the common ancestry. Then it can have internal nodes or divergence point that represent the hypothetical ancestors of the terminal nodes. And eventually we have the terminal nodes that you can observe right now, and these are represented as taxas. So the lines that join such terminal nodes, ancestral nodes, and the uh, root of your tree are termed as branches. Branches are sometimes also called as lineages. So this particular branch which arise from the root of the tree and has got one ancestral node that gave rise to terminal nodes A, we say that this is the lineage that is followed by this terminal node A, or this is the branch or lineage for the taxa A. That is what we represent in your phylogenetic tree. Okay, so there are some additional tree terminologies. Okay, the terminal nodes are also referred to as operational taxonomic units or something called as OTUs. These are also called as taxa. We have got internal nodes that I have already explained, and we have got terminal nodes. So one more thing that you should try to understand, that there would be branches, there would be roots, and the operational taxonomic unit or taxa that are most closely placed to each other in your phylogenetic tree are called as sister taxa. So if operational units are most closely placed to each other, then we can call them as sisters of each other. Okay, there is one more concept called as clade. Okay, the group of operational taxonomic units are usually referred to as clades. So this particular phylogenetic tree can be imagined to be formed of two clades. This would be clade 1, that is formed by operational taxonomic unit A, B and C. This would be clade 1, and the operational taxonomic unit D, E and F forms another clade called as clade 2, and since this clade 1 and clade 2 have a recent common ancestor, we say that this clade 1 is sister to clade 2. So clades can also be sister, and so can your operational taxonomic unit. So the clades or the operational taxonomic unit which has got recent common ancestors can be called as the sisters of each other. This is how you deal with different terminology, right? Okay, now one more thing that you should understand, that when you try to infer or when you try to understand any phylogenetic tree that is usually represented in a tree diagram that shows such evolutionary relationship among taxa, when you try to evaluate it, you should understand that there is no meaning as to what space is provided between the two taxa from top to bottom. Okay, or it doesn't really matter what is the order in which they appear in your phylogenetic tree from top to bottom. Okay, sometimes kids might misinterpret your results. Okay, so there is no meaning of if these two taxa are placed to each other. I mean, what is the distance between these two taxa? What is the space between these two taxa? That would not really matter. Okay, and usually whatever beautiful phylogenetic tree that you see in books and in your research paper, well, those are drawn using, say, some tree drawing software. Okay, so such tree drawing software take input your tree in this Newick format. Okay, so this Newick format is the format that is understood by your phylogenetic tree drawing software. Okay, and this Newick format are also sometimes referred to as New Hampshire tree format. In this Newick format, or this popular New Hampshire tree format, all the operational taxonomic unit are placed in parentheses, as well as they are separated by commas at specific location. Therefore, such type of data would be interpreted as this tree, and this tree would be drawn by your phylogenetic tree drawing software. Okay, and now how do we evaluate what is supposed to be shown in this tree? Okay, so this tree says that the operational taxonomic unit B and C are more closely related to each other as compared to A. Okay, and your taxa A, B and C forms a clade that is a sister group to a clade that is formed by taxonomic unit D and E. So there are two clades over here. This is clade 1, this is clade 2. Okay, so now if tree has the time scale which if it is provided with, then you can say that taxonomic unit D and E are most closely related, and so are B and C. So this is how you are supposed to interpret a phylogenetic tree. Okay, so now trees can be of two types. Trees can be either rooted or trees can be either unrooted. So over here, this picture or this part of your slide shows what a rooted tree would look like. The rooted tree, the root in the rooted tree usually denotes a common ancestor of all the operational taxonomic unit that you are comparing. On the other hand, your unrooted tree would only specify how the operational taxonomic units are related to each other, but it won't give you the evolutionary path in which these operational taxonomic unit have evolved. Okay, so your unrooted tree would only specify the degree of kinship among the taxa, but it would not give you an idea about the path in which these operational taxonomic units might have evolved. Okay, so in this way, based on if there is root in your tree, you can have two different type of trees, what we call them as rooted trees and unrooted trees. Okay, so now in order to infer evolutionary relationship between the taxa, Now it is important that you need to know how do you root your tree. Okay, so this rooting is usually done by mentally imagining that the tree is made up of strings, as you can see over here. So now what you are supposed to do is that you are supposed to grab the string at this root and tug it until the ends of the string, that is all these taxa, fall on the opposite side. This is what you are supposed to do. Imagine your tree is form of strings, decide a root and pull this root towards yourself unless and until all the taxa fall apart on the other side of your root. That is what is usually done when you root your phylogenetic tree. Okay, and in this case, we can say that we have rooted the tree with respect to A, and hence this A is usually termed as the out group. Okay, so you need to know the out group in order to root the tree. So all of the individual operational taxonomic unit except the out group now can be termed as in group. Okay, so in this way you have got two type of groups in your phylogenetic tree, what we call it as in group and what we call it as out group. Out group is used to root your phylogenetic tree. Okay, so this is what we were talking. We were talking about that out group. So this out group, that is we use some taxa, what we term them as out group, and these out group are known to fall outside the group of interest for which you are supposed to draw the evolutionary tree, and we call the group of interest as in group. Okay, so in this way some prior knowledge about the relationship among the taxa is required. It is essential that you have some basic background on as to how these taxa are related to each other. Okay, so the outgroup can either be species. Well, say for example, you can use bird as a root to root some mammalian operational taxonomic unit. Okay, well, say for example, if you are trying to find out relationship between mammals like donkey and lion and rabbit, then in order to root this tree, you can use this outgroup as the bird, right? As we know that birds are somehow different but related to the mammals. Okay, well, say for example, you can use some previous gene duplicate as an outgroup, right? So, well, say for example, if you are trying to build up uh, sorry, we are trying to build up a phylogenetic tree that involves beta globin from different species, that is beta globin from human, right, beta globin from some rodents, beta globin from marsupials, right? So if you are trying to build up a phylogenetic tree of such beta globins, then you can use some alpha globin to root those beta globin, right? So the thing is that you are supposed to know that this outgroup is somehow related to your in group, but it is but it has evolved differently. Okay, it is related but it is different. Such a kind of operational taxonomic unit are an ideal candidates to be used as outgroup. Just to give you an example, if you want to find out how stalks of winterfell are related to each other, then in order to root this phylogenetic tree, you would need another stalk that is somehow stalk, but it is not related directly to them. Okay, so you can use this Tony Stark to uh, out group the in group that would belong to all the Starks of Winterfell. So this is just a kind of hypothetical example. The idea is just to demonstrate how outgroup differ from in-group and what should be the properties of your outgroup. The outgroup should be related to your in-group, but you should have some idea that it is distinct from the members of your in-group. Okay. So we would talk about this outgroup and everything in more details when we do our practical demo. The things would become more clear as to how do we identify an outgroup. Okay, so now we are talking about some key concepts of evolution and proteins and the functional relationship among those proteins. So there are some key concepts that I think you should know, and these key concepts are very frequently discussed in your research papers or in your seminar. So it would be easier for you to understand what these people are saying when they refer to homologs, or when they refer to orthologs, paralogs and xenologs. Okay, so well, let us first try to understand the concept of homology. Okay, so homology has got a specific meaning. Say, if say some two genes are derived from a common ancestor, then we say that these two genes are homologs of each other. Well, say for example, we have got this gene 1 and you have got your another gene 2, and if this gene 1 and gene 2 share a common ancestry, that is they are derived from a common ancestor, then we say that this gene 1 is homologous to gene 2. Homology has got a specific meaning. You cannot call any two sequences as homologous to each other unless and until they have a common ancestor. Okay, so the genes or proteins which are derived from a common ancestors are supposed to be homologs of each other. And now based on how these homologs arise from the common ancestor, you have different type of homologs. We call them as orthologs, we call them as paralogs, we call them as xenologs. Okay, so homologs can be orthologs, homologs can be paralogs, or homologs can be xenologs as well. Okay, now let us try and understand what are orthologs, what are paralogs, and these are all types of homologs. Okay, homology has got a specific definition. If two genes or proteins are derived from common ancestor, then we say that these two genes or proteins are homologous to each other. If they do not share common ancestry, if they are not derived from a common ancestor, you cannot call such two or multiple genes as homologs of each other. Okay, so now let us talk about what are orthologs and how do they appear. Okay, so one can define orthologs as same set of genes performing the same function but in different species. That is, genes performing or the proteins performing same function but in different species are called as orthologs of each other. To give an example, hemoglobin in human is orthologous to hemoglobin in chimpanzee. Okay, since hemoglobin perform the same function, a gene that perform the same function but in different species are called as orthologs of each other. We would call them as orthologs of each other. So hemoglobin present in human is said to be orthologous to the hemoglobin that is present in chimpanzee. Okay, so this is how you define ortholog. Such orthologs are product of genetic events, or you can say evolutionary events like speciation. Okay, well, say long time ago, we had a common ancestor of human and chimpanzee. Okay, long time ago, we had common ancestor of humans and chimpanzee, and this common ancestor also has got this hemoglobin gene. But in due course of evolution, speciation event happened. Okay, due to speciation event, now two species have evolved from a common ancestor. We say, in our case, the two species, humans and chimpanzees, have evolved from common ancestor, and the same hemoglobin gene was retained in both the species that still perform the same function in two different species, right? And therefore we call such set of genes as orthologs to each other. Orthologs appear due to evolutionary events like speciation. Speciation give rise to orthologs, right? Now let us talk about what are paralogs. Okay, so one can define paralogs as a set of genes that perform different function but in same species. So genes performing different function in the same species are called as paralogs of each other. A typical example would be hemoglobin and myoglobin in human, right? So you have got hemoglobin gene and you have got myoglobin gene, right? And the two proteins or the two genes that perform different function in the same species are called as paralogs of each other. So these are called as paralogs of each other. Okay, so now let us talk about how these paralogs arise. Now we know that orthologs arise due to speciation event, right? While the orthologs are known to arise due to evolutionary events like gene duplication and gene mutation. Well, say for example, long time ago, there used to be an ancestral gene for globin. We would call it as globin, ancestral globin gene. Okay, so now this ancestral globin gene might have this gene might have been duplicated in say some due course of evolution, and we have got two copies of this ancestral globin gene. We would call it as copy 1 and copy 2. Now this copy 1 might have acquired mutations, and it might take up the function of transferring the oxygen, and hence we call this gene now as hemoglobin. On the other hand, another copy of the same ancestral gene might have acquired some independent mutation and took up the function of storing the oxygen, and hence we say that it has evolved into myoglobin. Now, since this hemoglobin and myoglobin were derived from a common ancestral gene, we say that these are homologs of each other, and since they perform different function but in the same species, we call them as paralogs of each other. So paralogs appear due to evolutionary events like gene duplication followed by mutation. So the genetic or evolutionary events like gene duplication and subsequent mutation give rise to paralogs, while the evolutionary events like speciation give rise to orthologs. Okay, orthologs perform the same function but in different species, while paralogs perform different function but in same species. So This is how we define what are orthologs and what are paralogs. Xenologs are the product of horizontal gene transfer, right? So if, well, say for example, uh, this orthologs and paralogs are product of vertical gene transfer, right? While xenologs are the homologs that are arise due to horizontal gene transfer that involve transferring of genes from one species to another species, right? So this is how xenologs appear. They appear due to the process of horizontal gene transfer. Okay, so today we have learned some key concepts like what are homologs, what are different type of homologs, what are different terminologies that are generally used when you discuss the phylogenetic tree. Now you know how do you interpret a phylogenetic tree, what is to be considered in your phylogenetic tree and what is not to be considered in interpreting your phylogenetic tree. If that is the thing that you have understood, well, the objective of creating this video or the lecture has been accomplished. Okay, so in our next lecture we would discuss what are different methods that are available for constructing your phylogenetic tree. Okay, so this is it what I have for you guys for time being. Okay, thank you for your kind attention.